DriverCheck. Medical testing and assessments. One phone number, one stop, one step ahead.
DriverCheck offers drug testing methods on a broad range of sample types, including urine, oral fluid, and hair. Each method has its advantages and disadvantages. This tutorial will help you compare them and will help you determine which type or types best match your company's objectives. For a lab-based urine collection, the sample is collected in a securely sealed specimen cup and is shipped to an accredited analytical laboratory for testing. As the gold standard model for workplace drug testing in North America, this is the only acceptable sample type for employers who are regulated by the U.S. Department of Transportation, or the DOT. The benefits? Urine testing generally provides a longer history of drug use than oral fluid testing. The longer time window of detection built into the process identifies risk and may therefore be preferred for risk-based programs. The downside? It is not an impairment test and may not be permitted for certain types of non-DOT regulated testing circumstances in Canada as a consequence of certain court, arbitration, and Human Rights Commission decisions. Further, urine testing is more susceptible to adulteration than is oral fluid or hair testing. For lab-based oral fluid testing, the sample is collected using a swab and is shipped to the laboratory for testing. Many clients use this for post-incident or post-accident, reasonable cause, and random testing. Depending on the positive test cutoff levels, analysis of this sample type may indicate the likelihood of drug use within hours prior to the collection and therefore could potentially indicate the likelihood of impairment during or in proximity to the work shift. Every test is directly observed, so there is less risk of adulteration or substitution. It is less invasive than urine testing. And the DOT is considering this test type for approval, but likely using longer time windows of use cutoff levels that will detect risk rather than the likelihood of impairment. The disadvantage of using test cutoff levels that reflect the likelihood of very recent drug use is that for risk-based programs, it does not provide a long history of drug use, meaning the identification of some intermittent or recreational drug users may be missed by using this testing method. And as stated before, a positive result may only indicate that the employee was likely impaired on the job. There are no guarantees. For urine point-of-care testing, the sample is essentially collected the same way as a lab-based urine collection, but using a container with built-in analytical capabilities. The sample in this case is tested or screened immediately on site, thereby eliminating the time needed to ship negative screening test specimens to the lab for analysis. That means it provides an initial screening test result within 5 minutes. The screening test cutoff levels match those used for lab-based testing for the standard 5 panel, and POCT kits can be considered reliable screening tools depending on company philosophy. Unfortunately, though, a urine POCT screen cannot be used to confirm a positive result. A lab confirmation test is required if the POCT screen test is non-negative. Also, the POCT is not as accurate as lab-based testing, so there is a higher risk of false positives and false negatives when comparing to the screening technology at the lab, though the technology has proven more reliable than oral fluid POCT. This test type is not yet approved by the DOT for its regulated testing program, and there are no industry standard cutoff levels for the drugs tested for beyond those included in the DOT's standard 5 panel. With oral fluid point-of-care testing, the sample is collected the same way as the lab-based oral fluid tests, but using a device capable of immediate screening on site. The test shows the screening result within 15 minutes of the collection, and every test is directly observed, so there is less risk of adulteration. The disadvantage is that the POCT test is an initial screening technology only, and in itself is not as accurate as initial screening technology used for lab-based testing. As with urine POCT, non-negative oral fluid POCT results must be sent to a lab for confirmation testing. And there's a greater chance of false negatives or false positives compared to lab-based screening tests, as well as currently available urine POCT tests. The other downside is that studies show these kits appear to have difficulty detecting drug use for some common drugs, most notably marijuana. At this time, we do not recommend the use of oral fluid POCT testing due to the disadvantages we noted. However, we continue to monitor this technology and will offer this product once it meets acceptable standards. Hair testing involves cutting a segment of hair from the head or body and submitting the specimen to a lab for analysis. It is most commonly used by organizations that require abstinence and risk-based drug testing programs. This is a valuable test because the standard 1.5 inches of hair can provide a 90-day history of drug use. Because of the longer detection time window and the fact that this collection is directly observed, the specimen cannot easily be compromised. Drug users cannot substitute or tamper with the sample or abstain for a few days and pass the test. Unfortunately, this type of test is more costly than an oral fluid or urine test. No POCT hair test is available at this time, and it cannot be used as an impairment test. For more information, visit DriverCheck.ca or call us toll-free at 1-800-463-4310.